So we now come to a very exciting part of today, perhaps the most exciting part. Young people are actually going to give lightning talks, 5-minute talks, no questions. This session is going to be chaired by Senjuti Saha, who is right now the leader in Bangladesh on child health research in a foundation called Child Health Research Foundation. She is pushing a Human Cell Atlas project in Bangladesh, collaborating with leaders of the HCA and with many of us. So we are very proud of Senjuti, and she's trained in the University of Toronto, has a very global view, and it's really my pleasure to invite you, Senjuti. Please come.
Shubho Shondha. I am Senjuti and I've come all the way from Bangladesh, as Dr. Partha was saying. And what I love about coming to Kolkata is that I come to a different country, foreign land, but I get to speak my mother tongue here. Shubho Shondha is Bangla for good evening. Shubho Shondha. That's your Bangla lesson for the day. I'll come back with another one tomorrow. Our first speaker for this lightning talk is Punn from Mahidol University. Punn, please come here. You have 5 minutes.
All right, thank you so much for this opportunity. So a very quick presentation to give an introduction to my poster. So no single cell details here. So if you'd like to see those details, please visit my poster. So today for this introduction, I'll be talking about the progress of pancreatic differentiations into islets from stem cells for type 1 diabetes cure. And so pancreatic islets contain these microtissues called the islet of Langerhans, and these things contain heterogeneous populations of different cell types. The most important one that we care about are the beta cells, which are responsible for producing insulin. And so insulin is responsible for reducing blood glucose, but when we have loss or dysfunction of the beta cells, you no longer have insulin. And so that resulted in high blood glucose levels, which then results to patients being diabetic. And so current strategies right now is really to inject with insulin. And so there's a lot of compliance involved and a lot of complications with this treatment. And so people have been trying to develop a permanent cure, and one of that is to do an islet allotransplant. But the problem with this method is that there's extreme limited supply. And so our goal here in my research is to find an alternative source for pancreatic islets. And so we've utilized some pluripotent stem cells, and then we would try to differentiate those into islet cells, islets of Langerhans that contain these insulin-producing beta cells. And so the goal of this whole research is to be able to make these beta cells and put it into diabetic patients so that we can restore normal glycemia in patients, type 1 diabetes patients. And so we've utilized a lot of different tools, particularly single-cell RNA sequencing, to get to this point where we developed a differentiation protocol to identify novel factors that could get us from stem cells into insulin-producing beta cells. And so if you were to differentiate stem cells into beta cells, this is the typical experiments or typical tasks that you would see into making these differentiations. And so the end result of this is that we get these organoids that are called stem cell-derived islets containing these insulin. And so we tested these organoids as a proof of concept to show if it would be able to cure diabetes in diabetic mice. And so we went ahead and got healthy mice, made them diabetic, and then transplanted these organoids to show that they are capable of reducing glucose levels in the blood of these mice. But the big question that often comes to us is that how similar are these stem cell-derived islets to the real human primary islets? And the question is The answer to this is that they're not at all similar. And one of the problems is that these SC islets here have different capabilities as mature adult primary islets. One of the things that you can see here is that SC islets don't have the capacity to secrete as much insulin as we want it to do. And the key problems are listed here. SC islets secrete less insulin, they're not mature, and they contain off-target cells, which to address all these three topics, we've utilized single-cell multi-omic sequencing, and that involves RNA sequencing and ATAC sequencing to address these issues. And so if you'd like to see those results, please, again, visit my poster, and thank you very much.
Thank you, Punn. You gave us 38 seconds back. Our next speaker is Kian Hong Kock from Genome Institute, Singapore.
Hello everyone, good evening. Thank you for the opportunity to speak today. My name is Kian Hong, and I'll be presenting on a lightning talk on my poster on the determinants of diversity in circulating immune cell states across Asia. So this is on behalf of the Asian Immune Diversity Atlas Network. As you have just heard from Carolyn in her fundus talk just now, as a community, we recognize that increasing diversity in genomics and genetics is a pressing equity issue. So you've heard the statistic a couple of times today, but though around 15% of the global population is of European ancestry, more than 85% of donors in genomics resources, such as the GWAS catalog or version 8 of the GTEx dataset, these are actually from European donors or donors of European ancestry. So in my poster presentation and my lightning talk, I'm going to focus on what we're missing out on with this particular landscape and what insights are possible from studying diverse population groups. So, supported by the Chan Zuckerberg Initiative Seed and Ancestry Networks, we launched the Asian Immune Diversity Atlas, or AIDA for short. The current version of the Asian Immune Diversity Atlas comprises more than 1.2 million circulating immune cells, or PBMCs, and these come from 619 Asian donors across 7 population groups in 5 countries. You can see this in the UMAP over here. We've annotated more than 30 cell types and subtypes and more than 50 cell identities. And please feel free to check out our cell by gene link over here, which contains version 1 of the human cell of our atlas. This particular atlas comprises individuals from Japan, South Korea, Singapore, Thailand, and also donors from here in Kolkata. And our study site over here is spearheaded by our wonderful HCA Asia host, Partha Majumder, and the lead postdoc, Ankita Chatterjee, whom you hear from later. So when we look at the broader AIDA cohort, we find that ancestry has a pervasive influence across multiple cell populations. And you can see some examples over here. For example, Singaporean Malay donors have a heightened proportion of IgHM low memory B cells. Thai donors have a lower proportion of CDC2 cells. And most strikingly, as Sean pointed out earlier, Korean donors have a lower proportion of Treg cells. So really, ancestry impacts cell type proportions across multiple cell populations. And we are able to leverage the single cell resolution of our atlas to identify differential abundance that's not immediately apparent at the level of cell types or cell subtypes. So for example, if you look at CD4 plus T cytotoxic cells, they don't differ too much in proportions across ancestries. However, if we look at, say, single cell resolution level analysis using algorithms like MILO-R, we find that, for example, in Singaporean Malay donors, there is a heightened level of a particular CD4 plus T cytotoxic cell neighborhood. And this particular neighborhood actually has heightened levels of cytotoxicity associated genes. So these are ancestry associated differential abundance that's really apparent at the level of cell neighborhoods rather than cell types or subtypes. So I'm not going to be able to talk about all the details, but if you come and visit us at our poster, we find that, and really human diversity it's not just really a pressing equity issue, but also is of fundamental biological importance. So from cell type proportions to variant effects, ancestry and human diversity really has pervasive impacts on fundamental biology. And we discuss cell type proportions, interactions between dimensions of human diversity, and some of the implications for healthy baseline levels of precision medicine. Going forward, we're hoping to expand this to the Ancestry Networks, so that's AIDA in blue over here, and also the Indonesian Ancestry Network project highlighted in red over here, as we work on expanding our knowledge of the impact of ancestral, genetic ancestry, environmental and geographical diversity on molecular and cellular variation. So in addition to my poster, we've got multiple posters from talented next generation researchers, including Lermin, Radhika, Eliora, and Yoshi, covering really a full spectrum of research from experimental benchmarking to cell type annotation to molecular variation and transcription factor activity and escape for X chromosome inactivation. So I encourage you to come and check out all of our posters over here at the session later. Finally, in closing, I'd like to thank my team at the Genome Institute of Singapore, as well as our AIDA partners. It's really been a privilege and a pleasure to be able to work with such talented and amazing colleagues. I'd also like to give a shout out to Jenny, I think, who's coming up next from the Lattice team, and to thank the Chan Zuckerberg Initiative for their support and funding our projects. So thank you for listening. I'm looking forward to meeting you at the posters.
You know, when I agreed to chair this session, Dr. Partha Majumder said, you're going to make a lot of enemies because you have to pull people off the stage. But everyone is taking so well to time. Next, we have Srimonta Gayen from Indian Institute of Science, Bangalore.
A very good evening to all. So it's been exciting to be here, and I'd like to thank the organizers for this opportunity. So let's begin. So I'll just change the little bit, shift the gear today. So I'll take you to the human early development. So what I'm going to do today, it's really long-standing question in what is the state of the X chromosomes in female human early embryos. It's remained till date a mystery. So I'll try to give some insight into that. So as you know, in female mammals during the early developments, among the two X chromosome, one X chromosome becomes inactivated to compensate the dosage of the sex chromosomes between the sexes. So, and if you know the mechanism very briefly here, so there is a very popular long non-coding RNA exists which nicely code the inactive X chromosome in female cells and recruits different series of chromatin modifier proteins to facilitate the heterochromatinizations of the inactive X chromosome. And now these X chromosome inactivation have been extensively studied in mouse systems. So in mouse during the early embryogenesis, so there is imprinted X inactivations, and then in these pluripotent naive embryonic stem cells, so this inactive X becomes reactivated, and then again in the primed epiblast stem cell in the post-implantation embryo, so it undergo random X chromosome inactivation. So this is very much well studied, but the state of the human pre-implantation embryos so remains still date a mystery. So there's different mixed outcomes. So there are so far two models. So first of all, in human there is no imprinted X inactivation. So as you know, in adult human you'll find the random X inactivations, but how it's initiated in the early development, so that remains different mixed outcome is there. Like some models here, so there are both X chromosomes, some sort of expression got dampened, and some other still like mouse, so there is inactivations of the one X chromosome. So we took the advantage of the human pluripotent stem cell to address this issue. So but the conventional human pluripotent stem cells represent actually the prime state, which is already undergoing the inactivation, does not represent this pre-implantation pluripotent epiblast precursor cells. So what we used, what we did, so we converted these prime cells to the naive state to revert back to the pre-implantation stages and try to ask that what is the state of the X chromosome in these naive cells using the single cell RNA-seq analysis. So in very briefly, what we found that if you see the early naive cells to late naive cells, so if you look at the exist expression, so their early naive cells almost didn't have no exist expression, but it comes back at the late cell. And what surprisingly also we see that early to late there is huge down regulation of the X-linked gene expression. So now we wanted to see that whether this down regulation of X-linked gene expression because of the X inactivation or dampening. And surprisingly, so because of the lack of time, I'll not go through, I'll just skip these. So what we found, that there's no inactivations of the X chromosome. Here the red color representing the X gene state. These two colors different monoallelic state. So as you can see in the both early and late naive cells, so there are a lot of biallelic gene expression. So which is indicating there is no X inactivations in these cells. So next we also looked at is there a dampening probability. So we compare the biallelic gene expression from early to late naive cells. We don't see any significant changes. So then we move to that, what could be the region of the downregulation of gene expression from early to late naive state. So recently we shown in the mouse that there is a concomitant upregulation of the active X chromosome during the early embryogenesis upon inactivations of the X chromosome, and when the inactive X gets reactivates also, there is erasure of the upregulation. So we wanted to see if same thing happens in the human prime to naive conversion. So to the model, what we really see that human prime cells, it's really when you have inactive X, other X chromosome is upregulated, and when you are converting to the early or late naive state, so there is the erasure of upregulation is happening as we described in the mouse. So that is actually leading to the downregulation of X-linked gene expression during the prime to naive conversion. So with these, to summarize this, so what do we think? There is no inactivation or dampening in the human naive pluripotent stem cells, and there are a lot of heterogeneity. So it's also there are some genes still remain inactivated, and many genes still remain as active state from the inactive X chromosome. So with this, I'll the end of my talk, and I want to acknowledge the funding agency and the two graduate students who contributed to this work, Susmita and Deepsika, and thank you for your patience.
Thank you. Next, we have Edahiro from Osaka University.
I appreciate the opportunity to present here. My topic is about integrative analysis of single-cell data and hostogenesis data in COVID-19. COVID-19 GBAS has demonstrated that the hostogenetic background influences the susceptibility and or severity of COVID-19. However, the functional analysis of the GBAS identified genetic variant has mostly focused on the variant on chromosome 3, which have a strongest severity association in European, but confer allele frequency is Asian, suggesting the importance of the additional study in non-European. Therefore, we aim to elucidate the immune response in COVID-19 from both a pathophysiological and hostogenetic perspective in the Japanese population. This is an overview of a study. We recruited 75 healthy control and 73 COVID-19 patients at Osaka University in Japan. The PBMC from this subject, we performed 5' single-cell RNA-seq, single-cell VD-seq, and SNP genotyping. And this is a UMAP embedding of all PBMC, and cell counts are around 1 million. To evaluate the genome-wide host risk of COVID-19 and identify the disease-relevant cell type, we integrated our single-cell data and the COVID-19 GBAS of 3 phenotype from a COVID-19 HGI round 6. First, we identified the top 100 putative disease gene of each phenotypes. And we computed a disease score at single-cell resolution of 3 phenotypes. And also performed the cell type enrichment analysis, such as association and heterogeneity using HCDRS. These are the UMAP embedding of PBMC colored by disease score. Disease score are similar across cell type when GBAS cases were cell-reported COVID-19. However, the cell annotated as monocyte and DC have a higher disease score than the other cell type when GBAS cases were restricted to hospitalized or very severe one. Next, we perform the cell type enrichment analysis. First, we assess the association between 3 phenotypes, and major 6 cell types. And we found that no cell type enrichment in cell-reputed GBAS. However, monocyte and DC have a significant association with hospitalized and very severe one. In addition, 3 cell types have a significant heterogeneity in association with severe COVID-19 phenotypes. Next, we investigated the monocyte and DC cluster and found that all of the 5 subsets have an association with severe phenotypes, which was more prominent in the very severe one. This data suggested that polygenic risk involved in COVID-19 severity enriched in innate immune cells. To gain a better understanding of transcriptional variability and dynamics regulated by the COVID-19 risk variant, we performed single-cell-based EQT analysis. We examined 5 variants, which were replicated in Japanese COVID-19 GBAS out of 13 risk variants identified at the first round of the COVID-19 HCA GBAS. At first, we performed the single-cell-based EQT analysis at the major 6 cell types separated for COVID-19 and healthy. These risk variants show distinct different cell type enrichment pattern with significant EQT effects, suggesting context-specific EQT effect. Especially, IF-RNA2 variant have a COVID-19 and monocyte-specific EQT effect. Therefore, we investigated monocyte cluster in more detail and found that the IF-RNA2 variant have a classical monocyte-specific EQT effect in COVID-19, and no such EQT effect was observed in healthy. IF-RNA2 have a key role in type 1 interferon signaling pathway. Therefore, we investigated this variant in more detail. This is a box plot of the EQT effect of the IF-RNA2 variant in total monocyte and classical monocyte. And we found that risk RL T have a COVID-19 context-specific increasing dosage effect and a significant interaction between COVID-19 and healthy. And differential expression analysis also revealed that IF-RNA2 expression was increased in classical monocyte or COVID-19. The data suggested that the risk RL might contribute to severe COVID-19 by increasing expression of IF-RNA2 in classical monocyte. In summary, this study revealed that the hospital risk of COVID-19 severity was enriched in innate immune cell. Also implies that the integrative analysis of single-cell data and hostogenic data could aid in understanding the etiology of disease and the potential for personalized therapy. Finally, this study was co-supervised by Professor Okada and Professor Kumanogo, and co-led by Dr. Shirai. I really thank all the participants involved in this study. Thank you.
Our next lightning speaker is Jennifer Chien from Stanford University.
Thank you. I'm happy to be here today. My name is Jenny, and I'm a data curator for Cell by Gene. Cell by Gene contains a repository of standardized open source single cell data, currently at 59 million cells across 1,100 data sets. All the cells in the data corpus conform to the Cell by Gene schema, and the goal of the schema is to enable data reuse and integration while remaining minimal and concise. The schema leverages ontologies for things such as cell types, assays, and tissues, and gene features are indexed on ensemble IDs. All raw counts are required, while normalized counts are highly encouraged. And this schema is the minimal requirements, but we also definitely encourage any additional metadata that will help explain and give your experiment some context. The H5AD is systematically validated against the schema upon upload to the portal, and the data corpus is also periodically migrated to incorporate schema changes and ontology updates. And so the schema has enabled a suite of tools that Cell by Gene Discover. And whether you're a bench scientist or a computational biologist, there's a tool that will enable you to explore and analyze the data corpus. The collections and datasets page allows you to search on metadata such as disease, cell type, and tissue. And downloads of the individual datasets are available in both Seurat and AIDA. At the Explorer, users are able to look at individual data sets in more detail, and you could color by metadata or genes of interest. You can also do a differential expression on your selected cell subsets. Gene expression is a dot plot that enables you to quickly visualize expressions of genes across data sets while filtering and grouping by specified metadata. And last but not least, the census is a Python and R API that efficiently queries 40 million cloud-native human and mouse cells, regardless of the data set of origin. It's a tool that allows you to, for example, gather metadata and gene expression of all macrophages across all the data sets. So using the census, we're able to see the distribution of self-reported ethnicity of healthy human cells in the corpus. And this is one of many of the similar graphs that we've shown today. And this is very similar, except we have 49% unknown and 38% of European origin. And earlier this year, CZI announced that they'll be investing in AI for the sciences. And the data corpus at Cell by Gene will be part of the training data that feeds into these future machine learning models. So it's important that we expand and grow its diversity. And we're very thankful for efforts like AIDA that have made a difference in increasing representation from Asian communities. So if you're interested in contributing data, this is the general process. You can email us at cellbyGene@chanzuckerberg.com. As long as your data meets requirements, we will give you guidance and assist you in formatting your H5AD to the Cell by Gene schema. We will then create a private collection for you that has a stable URL which you can share with collaborators or reviewers. And once you're ready to make the data public, we will publish the collection for you. So I want to thank the curation team at Stanford, known as Lattice, with our PI Mike Cherry and group lead Jason Hilton. And we work closely with the amazing folks at CZI Single Cell Team and Clever Canary. And we appreciate our funders, Chan Zuckerberg Initiative, and a big thanks to all the collaborators and contributors that have worked with us to make this possible. We've recently come out with a preprint, and that goes into all the details. And please come find me anytime. I can demo stuff on my laptop.
Thanks, Jennifer. Next, we have Ankita Chatterjee from John Morton Center for Liver and Innovation.
Hello, good evening. Today I will be talking on the effects of smoking on peripheral immune system among healthy young adults. Now, we all know that smoking has a serious effect on host immune system, but in India we see that most of the adult users of tobacco starts using tobacco in the childhood or adolescence. It's so much serious that among these male school children, 6.9 to 22.5% practice smoking, but it's comparatively lower among the girls. Now, we also know from previous study that chronic cigarette smoking induces chronic inflammation, compromises host immune system, and causes immune suppression. Tobacco, the chemical composition of tobacco contains, along with increased level of carbon monoxide, can affect immune cells' development, growth, and functionality. And with this background, we hypothesize that smoking alters antigenic exposure to peripheral T and B cells, thus inducing class switching and altered immune repertoire in the peripheral circulation. We have included a cohort of 14 young young people, aged between 20 to 35 years. Among them, 7 were smokers, which is defined by the consumption of at least 3 cigarettes per day for the last 6 months, and 7 were non-smokers. From these 14 individuals, we collected blood samples, isolated PBMCs, we have profiled the single cell gene expression and also the TCR and BCR. For the immune repertoire profiling, we did the analysis using SC repertoire, where we specifically looked at the CD3 distribution. We called the clonotype, we look at the CDR3 motif, repertoire comparison, and clonotype tracking. So the first thing what we did is we looked at the clustering from the approximately 23,000 cells obtained from these 14 individuals, and we could identify major cell types along with cell states, and each of these cell types and cell state cluster is uniformly represented by the 14 individuals. We next compared the proportions of cell types and cell states, and the boxed cell types and cell states showed significant differences, specifically mentioning the increased proportions of B cells and decreased proportions of CD8 positive T cells. This work has been done by my junior Supratim Ghosh, who is also presenting a poster. Please, you can go and visit. We next then looked at the clonotype abundance, and we looked specifically for the TCR and BCR, and we did see that cell-specific clonotype is expressed. We did identify cell-specific clonotypes, but most of these clonotypes are present in single copy. Only few of them are present in multiple copies. We looked at the diversity index using the Shannon diversity index and inverse Simpson diversity index, and we did find reduced TCR diversity among the smokers compared to the non-smokers, but increased BCR diversity among the smokers compared to the non-smokers. We then looked at the common and unique clonotypes, and we found more common clonotypes among the BCRs rather than the TCRs between the smokers and the non-smokers. We used Morista index to look at this commonality, which is an estimate between how much a clonotype is similar across the individuals, and it ranges from 0 to 1. Here the more yellow color is towards 1, and as you can see that across individuals BCRs are more shared but not the TCRs. We also performed clonotype tracking to see how much clonotypes are actually shared between the non-smokers and smokers. As you can see, it's all again showing the same thing, where the BCRs have more similar clonotypes compared to the non-smokers, but not in the TCRs. Also, the proportions of each clonotype varies among the non-smokers and the smokers. Among the uncommon clonotypes, we found that most of the clonotypes are present in singlets. Only some of the unique clonotypes are present in high copies for the BCRs, and also mainly for the BCRs and also for TCRs. So to conclude, we did obtain higher unique BCR clonotypes, but lower unique TCR clonotypes among smokers. The proportions of each of the common clonotypes varied between the smokers and non-smokers. Few had high abundance, but most of them are present in single copy. And the diversity of the TCR was also found to be lower among the smokers compared to the non-smokers. So this is only this is a kind of a pilot study where we have taken a small group of individuals, and I would like to thank the study participants who have given consent to collect the samples. The whole thing has been initiated as a part of Asian Immune Diversity Atlas Network, and I would like to thank the network for helping me in designing the experiment and also doing the analysis. I would like to thank Human Cell Atlas for letting me speak in this platform, CZI for funding, National Institute of Biomedical Genomics, where the work has been done, specifically thanking Orinder Moitro, Supratim Ghosh, Sumanta Sarkar, and Silarim Kurkalang, and finally, my institute, John C. Martin Center for Liver Research and Innovation, with special thanks to my supervisor, Partha Majumder. Thank you.
Thank you, Ankita. And that marks the end of the lightning talks. There is no question-answer part to this session, but that's what the social hour is for, and please do visit the posters. Thank you.
All right, it's the end. Make it really short. Thanks everyone for staying to this late. I just want to say it was really a fantastic meeting. I think there's a lot of discussions, great breakout discussions. It's going to continue tomorrow. There'll be more talks related to genetic diversity. There'll be cancer talks, computational talks. So please come back. Especially we'll start at 9 a.m. With a very interesting talk. I won't say who it is. Anyway, just kidding. Please be here on time, and we're gonna have a fantastic day tomorrow. And please join us at the social event, and let's continue our discussion and potential collaborations. Partha, you wanna say something?
Thanks, Jay. Thanks everybody for being here and for those wonderful lightning talks. Housekeeping, we take a 30-minute break, come back here for beverages and food. Thank you very much. Look forward to seeing you tomorrow.